Okay, let's look at the answer. I hope after going through the structures of both collagen and DNA, right, the answers will be more understandable. Okay, it's a compare question. Okay, so you should be talking about both similarities and differences. Some of you, the similarities are not clearly shown. I need to find the similarities among the differences for you. So you downright forgot or did not even think about putting down similarities. Okay, so this time around, this is your first exam, so I did the finding of the similarities for some of you. Okay, next time no more. And I can assure you in the A levels they won't do that. Okay, so suggestion is first of all, of course, you must remember to put down the similarities, and secondly, separate your answers into paragraphs, right? So you can have one paragraph on similarities on its own, so that it's more easily seen, right, by the marker. Okay. Also, right, the question is on structure only. Okay, so for the many of you who mentioned things like function, things like solubility, which is a property, right? Things like where you can find the DNA and the collagen, all these are not structures. Right? You are just wasting your time writing things that cannot be awarded, no matter how correct they are. Right? So read the answer carefully, read the question carefully, and follow what the question is asking for. Follow the boundary, right, set by the question. Okay, so for the similarities, okay, I don't think I need to explain too much. Right? They are both made up of many monomers joined together to form polymer chains, and the joining of the monomers are via covalent bonds. Of course, the monomers are different, the covalent bonds are different, but the idea that they are both polymers formed by covalently joined monomers, these are similarities. Okay, and if you went back to the structure, right, it shows you that each of the chain, either it is a polypeptide chain or the DNA strand, both form helixes, and both the collagen and the DNA molecule, right, contains more than one chains, right? For collagen, it is many polypeptides, three in one triple collagen, then aggregated into fibril and fiber, so many polypeptides versus two DNA strands in one DNA molecule, right? So multiple strands or chains, right? And between all these polymer chains, there are hydrogen bonds that stabilize the chains together. Okay, and both are overall fibrous, meaning both are overall linear, long, right? Same shape, even though they are helical, right? Helical and fibrous are not mutually exclusive. Helical and linear also not mutually exclusive. You can be fibrous and helical. Okay, so some of you took that as a difference, right? Some of you mentioned collagen is fibrous versus DNA is helical. That is really not a difference. Okay, move on to the differences. Right, you can talk about the presence of different elements, but if you do that, I would expect all the elements to be shown. Right, so for collagen, because of the amino acids, where you have carbon, nitrogen, oxygen, hydrogen. Okay, I'm not going to mention sulfur because I know that there are no disulfide bonds in collagen, meaning there are no cysteine amino acids which contains the sulfur. Right, so really just CNOH. For DNA, there's an extra phosphorus because of the phosphate group in the DNA nucleotide. Right, the element is phosphorus, the element is not phosphate. Then the monomer, right, amino acids versus DNA nucleotides, there shouldn't be an issue. Then the modifications of monomers. So in the collagen, you know that there are modified amino acids, but there's no such thing as modified DNA nucleotides. So you can take that as a difference. Then how the monomers are joined together into a single chain, right? So there is bond between monomer. Okay, before I continue, you look at the feature, right? Your feature needs to be clear. If you want to write point form or just write 1 to 2 words in your body of the table, right? So if your feature is not clear, then you need to have more details. For example, right, for bond, for example, you just wrote bond, but bond can be between different things, right? Are you talking about bond between monomers or bond between chains, right? So you need to be more clear. What level of bonding are we talking about, right? So bond between monomer, that will be very clear. Then that will be peptide versus the phosphodiester bond. For the repeating sequence, right? Obviously, throughout the polypeptide of collagen, there is this repeating sequence of glycine, usually proline, and usually hydroxyproline. So in the previous video, I mentioned usually, not always, right? So not only these three amino acids. Okay, so for those who mentioned that in your answer, so it's glycine, then usually proline, usually hydroxyproline. In the DNA, some you mentioned there's no repeating sequences. That's not true. There are repeating sequences in certain parts of the DNA. So unlike collagen, where it is repetitive throughout the entire polypeptide, in one DNA molecule, right? Parts of the DNA molecules will be repetitive, like your telomeres. So you mentioned the centromeres as well. That is also true. Okay, but not the entire DNA molecule. But certainly there are repeating sequences. Okay, then for the next one, you can talk about directionality of each strand or each chain. Right, so each polypeptide chain has got an N terminus and the C terminus. For each DNA strand, there's the 5' and 3' end. For the number of chains, here we are talking about individual chains coiling together, right? So it is really a comparison between just tropocollagen rather than the entire collagen. It's just tropocollagen, which involves the coiling of individual chains. Okay, that would be a more fair comparison versus the DNA molecule, again formed by the coiling of individual chains, right? So more fair in terms of a feature of comparison. Okay, so it's just tropocollagen versus DNA molecule, therefore 3 polypeptides forming triple helix versus 2 DNA strands rather forming double helix. Okay, then for the further aggregation, now you see I separate the aggregation into fibrils and fibers into a different comparison rather than compare this to the DNA double helix. Okay, so further aggregation after we have formed the tropocollagen that is present, of course, in the collagen, as I mentioned already, fibrils and fibers. But between DNA molecules, there's no more aggregation, right? You don't see different DNA molecules coming together anymore, right? So no further aggregation. For the point 9, the hydrogen bonds formation. So we know that it is a similarity that there are hydrogen bonds formed between individual strands, right? But where and how they are formed is different. For collagen, that's how you saw in the structure already, when we went through the structure again, right? The hydrogen bonds are found within a tropocollagen, right? Found between adjacent polypeptides within a tropocollagen and formed not by our groups, right? Formed by the carbonyl and amino group of amino acids on adjacent polypeptides. Okay, then for the hydrogen bonds, also called complementary base pairing for DNA molecule, right? That those are formed between complementary bases of DNA nucleotides on adjacent DNA strands. Okay, so while you look at the difference, I also want you to look at how do we group the differences together. You can see that the differences has parallel, right, between the collagen and the DNA. We are talking about here specifically hydrogen bonds, specifically bonds between individual strands or individual chains, right? So that's the parallel. Then that gives you a better feature of comparison versus some of your comparisons. There's no parallel at all, right? You would compare, say, for example, peptide bond versus hydrogen bonds in DNA. What's the similarity or what's the parallel, right, in terms of the feature of comparison? Don't have, right? So the choice of your feature of comparison, the choice of your contrasting features, right, needs to be valid, needs to be well thought of also, right? It's not just randomly or bond versus bond, but different levels of bonding. That is not comparable. Okay, so I sidetrack a bit. So back to this answer. Okay, now point 10. Okay, other bonds. Right, so we know that there are, because of the presence of aggregation, right, the tropocollagens are aggregated together. There are covalent bonds or covalent cross linkages between the tropocollagens, right, formed between the alkyl lysine residues. But DNA has no further aggregation, so therefore also no other bonds joining the DNA molecules together. Okay, then the coiling, right? Collagen is not coiled around anything, but the DNA molecule is coiled around histones to form nucleosomes. Then there is further coiling. Of course, I'm not going to go through the details here, right? Just a simple comparison of presence and absence of coiling, right? So coiled around histones, further coiling into chromatin or chromosomes. Okay, then for the orientation of your individual strands or individual chains, right? So again, we are talking about between tropocollagen and DNA molecule. Okay, not your collagen fibril, not your collagen fiber. Why? Because those are formed not by individual polypeptides, right? Those are formed by tropocollagens aggregating together, fibers, rather collagen fibrils aggregating together, right? So when we look at one tropocollagen, you cannot say direction, right? Direction is for individual polypeptides, individual DNA strands. Okay, so when we talk about orientation, anti-parallel, parallel, we need to talk about individual polypeptides, individual DNA strands. Okay, so when we are looking at individual polypeptides, then of course my level I have to look at tropocollagens. Right, I'm comparing between the polypeptides within the tropocollagen, and they are parallel. Versus comparing the two DNA strands in the DNA molecule, they are anti-parallel. Okay, so you get. I hope you understand why the comparison here is between tropocollagen and DNA molecules, not between collagen fiber and DNA molecules, for example, because that's what some of you have tried to do. All right. Okay. Next, shape. Right. Some of you mentioned that the prokaryotic DNA molecules can be circular, not fibrous, not linear. Good. That's a good point. Right. That is an extra point that I accepted because while the question did not mention specifically whether the DNA is eukaryotic or prokaryotic, right, because collagen is only found in eukaryotes, so I assumed that the context of the question is eukaryotic DNA. But that's an assumption, right? So if you point out specifically that there can be prokaryotic DNA that are circular, that's a good point. So I accept it as an extra point, and AVP, which means any other valid points. All right. Then for the charge as well, right? I did not included this in my original answer because I do not know exactly all the amino acid makeup of the collagen. I remember collagen, besides the three that are usually present, there are other amino acids as well, right? And who knows? Right? Some of them may be charged. So I don't know. So I don't know whether I can say that collagen is 100% uncharged or neutral, but then since most of the amino acids in collagen that we know of are not charged, okay, so I accepted this as a point also. It should be overall close to, if not neutral, okay, versus DNA molecule are negatively charged. So I accepted this as an extra point as well, while it was not in my original answer. Okay, so therefore, again, AVP. Okay, so in order to score full marks, right, this is a 15-mark question, and this is how essays will be like for your A-level as well, right? For a 15-mark question, 10-mark question, whatever number of marks, there's always 1 mark that is reserved for quality of answer, okay, which we call QWC, all right? So in this case, because it's a comparison question, the quality of answer will be about the structure of your answer, whether you included both similarities and differences. So I'm looking for at least 1 to make it easier for you, because some of you only have like 1 similarity that I have to find for you, right? In the A-level, it may not be just 1, yeah? In the A-level, they can ask for, or they won't tell you, but the marker will maybe look for, must have 3 similarities, 3 differences, for example, to score this 1 mark, okay? But in this, for today, right, for this exam, right, just 1 similarities, 1 difference, and your differences are written in the point-to-point structure, okay? So if you fulfill all this, I'll give you that 1 mark. So that's the answer for question 5A.